we're going to be looking at how the technology can support you in terms of monitoring outbreaks and through various papers that have been published, and we'll be looking at some examples of these. Okay. So just as a tiny brief review, my recommendation is, if you haven't yet watched the introduction to ddPCR, to please watch that video. That'll be, I think that was carried out on the 25th of July, if I'm not mistaken. You'll be seeing me again presenting on that, but that was just introducing the technology, gives you a nice sort of broad overview and good understanding of where it sort of fits and how it works. I think as a scientist, it's always important to understand the technique so you're able to apply it better. So I'm not going to go into detail this afternoon, just simply highlighting the fact that ddPCR is what we describe as the third generation PCR. Where it moves, it's sort of a combination of endpoint and real-time PCR. It's a endpoint in the fact that the droplets or the partitions are thermal cycled to the end, where they are looked at only at the end of the cycling process. But they are real-time, similar to real-time in the fact that they are using fluorescent dyes to monitor them. So that's why we say it's kind of a combination of both. But the big differentiating factor is that they use partitioning as a means of dividing the droplets, or putting those droplets, and in that so doing, it allows a counting or quantification method by counting, and that's where the digital part of digital PCR comes in. So the workflow should be semi-familiar to anyone that's doing any sort of PCR. The only difference is in the fact that you're doing a droplet generation step. That droplet generation step takes each well of your 96 well plate, and it just divides it into around 20,000 partitions per well. And what that means is your reaction is divided up so that every single little partition or droplet is PCR occurs on each single droplet. So basically, I like to think of it as having 20,000 PCRs occurring instead of just one in each well. So once you've done the droplet generation, you move it to the amplification. That's a thermal cycler. And then you do your droplet reading step in a droplet reading system, and that's done on the 96 well plate. In that system, it picks up all your droplets that have been thermal cycled, and it sort of singulates them like a flow cytometer, and then it reads each droplet one by one. So it just gives a good understanding of how the process works. Now, what does that mean for the technology? Well, it means that it does have a high sensitivity for targeted analysis. It offers much higher sensitivity when then compared to things like NGS, because by partitioning the sample into thousands of individual droplets, ddPCR is able to detect much more limited rare mutations or low abundance targets with greater precision. It also has a much faster turnaround time than other similar technologies. It does involve things like library preparation and sequencing, as well as data analysis, which can take a long time. But most importantly with ddPCR, especially in today's sort of discussion, is the fact that it's an absolute quantification method. So what do we mean when we say it's an absolute quantification method? We mean that it gives you a quantity of your unknown sample or your gene of interest, and that's given in copies per microliter. And that concentration or quantification is done without the need of a standard curve. Whereas in real-time PCR, it's a relative quantification method. You have to compare to an unknown to a known, and then in so doing, you can get an idea of where you are in terms of quantification. Whereas in digital PCR, it's a counting method, and it gives you copies per microliter. And then really today, another two big focus points is going to be the sensitivity, a greater sensitivity when compared to other PCR methods, and much higher, like I discussed, in things like NGS, and then inhibitor tolerance as well, and how that can sort of increase your quality of results and things like that. So this is a very classic picture. If you've seen anything to do with digital PCR, it's called the needle in the haystack approach. And basically what it means, it just means if you were looking over a haystack with a needle in it and you had a metal detector, you were waving the metal detector over that haystack, you could definitely tell that there's a needle in there because your metal detector would go off, but you can't tell if there's one needle or 500 needles. And you'd have to count each needle with a metal detector separately. So if I divided that haystack into around 20,000 smaller haystacks, and I put the needle in one of them, and I gave you that same detector, yes, you'd have to walk or move around in some space, but you'd absolutely be able to detect how many needles and exactly where there are as well. So that partitioning effect really increases the number of decreases the background molecules that you're looking against for your molecules of interest. In this example, you're looking at in qPCR, PCR, you're looking at 40 mutant molecules in 40,000 wild types, which is just a 0.1% abundance. And when you compare that to ddPCR, you're looking at a 33% abundance because it's just a single droplet is either counted as positive or negative. So it's just a chance of picking up that single droplet, and that chance of it being detected is a 33% chance. So really that titrating out is a big assistance to detecting a limited quantity. So these are just some of the applications that ddPCR is really useful for and really helpful in terms of monitoring. But we're going to really focus in on two of the largest ones when it comes to surveillance, surveillance and disease monitoring. And those two are wastewater detection, which has really taken off since COVID and has actually increased quite a lot in terms of the monitoring that's been done with that. And then viral load, viral titer, and then obviously this isn't part of the supporting challenging applications, but just detection, presence, absence as well. In terms of challenging applications, obviously the field is quite large, and this is just a small sort of subsampling of the different applications that can be used by the technology. So really just an overview. But we want to get into the more detailed outbreak surveillance. How can you use it if you're trying to do outbreak surveillance monitoring? Well, I said to you, we're going to be looking at wastewater-based epidemiology. So that's termed now WBE, that's wastewater-based epidemiology. And how can ddPCR be used for something like this? Well, firstly, let's just look at the importance of surveillance. And when we're talking surveillance, we're talking both disease and wastewater, just any disease. And the importance is that five-stage process that we've taken off the CDC website: detecting a disease where and when it happens, stopping disease before it spreads, to study a disease and to strengthen the science behind it, improve how we can prevent and control diseases, and to protect populations and keep people healthy in the long run. That's really what the focus is of today's sort of webinar, and how ddPCR can enable that. Now, if we look at wastewater-based epidemiology, it's really utilized to give a comprehensive health information on a community. So the concept is primarily based upon the extraction of a sample from wastewater, and then analyzing and interpreting that samples, either some sort of chemical or biological compound in there, and these are referred to as a biomarker. Now, in the sense of specifically with ddPCR, we're mostly looking for genomic biomarkers, that's DNA or RNA that's present in the water, so nucleic acids. And most of the time, these can be used to sort of monitor a whole community. Why is that more helpful than monitoring patients that report to a pathology lab? Well, the first is you have a much bigger data set. Often in poorer areas, you may not get people that are able to go to a pathology center, and that means you won't actually detect it. It also, often you will see a big spike in wastewater well before you'll start to see an increase at pathology labs, just because of the delay in people going to get tested from once they're starting to show symptoms. And also you can use it for monitoring things like institutes, maybe universities, or some specific outbreak in specific areas, and knowing what to do to be able to monitor and to treat them. So why do we recommend ddPCR for WBE, for wastewater-based epidemiology? Well, we know that ddPCR, why it is used, and we know why where it can benefit over existing technologies such as real-time PCR. But let's break it down to look at each specific point. So the most important reason why one would use ddPCR over real-time PCR is you don't have to use standard curves. So I've taken a publication that looked at the danger of using a CQ value to quantify nucleic acid in biological samples, where they actually looked at the error that they were getting from using RT-qPCR. And while it was really instrumental in response to COVID-19, they didn't recommend using CQ or cycling threshold or crossing point values to set clinical cutoffs. They said this was due to poor interlaboratory reproducibility, and they said rather to recommend using calibrated copy-based units that are used elsewhere in virology, such as those in ddPCR, which use a copies per microliter results. So the one big benefit, why, is the no standard curve, because it's an absolute quantification method. It's much more inhibitor tolerant compared to qPCR. It's reproducible. It's got a quick turnaround time, and these are just some of the small benefits, or rather some of the benefits that just, when added together, make a huge difference in actual monitoring of patient samples. So something that's quite common, or something that's really becoming more common these days, is human monkeypox. So commonly, the way that this is being monitored is by swabbing of a lesion, so a lesion on the patient, and you collect samples for testing. However, by this stage, infection is already well established and likely spread by the time these lesions are appearing. So wastewater-based epidemiology is a really good passive collection method, and we know that it's worked really well with SARS-CoV-2, and it can proactively detect virus in a community around and supports powerful longitudinal trend studies over time. Now, if we compare it to existing technologies, there's obviously pros and cons to each method of the current testing methods. So the first cultures, or microscopy, electron microscopy, they are low throughput and they take nearly a month to obtain your results, and they require highly specialized equipment. When you look at rapid diagnostic techniques such as ELISAs, they can detect serological antibodies or viral antigens with moderate sensitivity, but cross-react with other viruses that may be present. qPCR obviously started to emerge as a strong tool in coronavirus, but as we saw, there was limitations with CQ settings. The results were not always accurate, and this is really where ddPCR is able to pick up, especially when we're looking at low viral copy number that's often unreliable by qPCR. So when we looked at the COVID pandemic, WBE proved and continues to prove effective for really good longitudinal detection and predicting of community surges for disease outbreak. So in addition to circumventing or the other, trying to get around these other sample collection methods, ddPCR offers a much higher sensitivity with absolute quantification methods. And this doing really helps much more actionable results for patients as well. So in the slide, you'll see we actually have a kit that's been validated for this monitoring in wastewater. And this sort of ties in nicely to the next slide, which just highlights some of the kits that we're currently offering for wastewater surveillance. So some of the assays that we're offering. And as you can see there, some of these assays already gotten quite large in terms of the panel size. which is a 6-plex assay with adenovirus, norovirus, influenza A and B, and 2 monkeypox clades. And then those two, over time, are we continuing to build much larger panels in collaboration with the US, the CDC, and the wastewater surveillance team there. They're looking at building panels of up to 30 different analytes at once. So really a great future for wastewater monitoring and just sort of community monitoring in terms of disease outbreak as well, and those sort of technologies. Now we're going to look at surveillance in terms of our pathogen detection with ddPCR. So when we talk about pathogen detection obviously is quite a broad topic, as we can see by this paper in 2007. You've got defense as one area, the food industry, clinical, water and environmental samples, as we kind of touched on with wastewater-based epidemiology and other areas. So when we discuss with customers, obviously what they're expecting and what we're trying to offer can be different. But mostly we're looking at clinical to a degree with water, environmental, and some of the food industry. But just to touch on some of these this afternoon, we want to look at just a few of them. So if we look at some of the current approaches to pathogen surveillance, you've got obviously your culturing method, which you grow pathogens on a culture dish or like a liquid media, and you do colony counting or optical measurements. But the problem is with viable or non-cultural bacteria that can cause issues. You can also do something like a blood smear, which is an optical observation under a microscope with patient blood samples. And you can use that, but obviously that has a lot lower accuracy. You've got serology, which is the bodily fluid study, and you're looking for a specific antibodies in the system. And you've got your one that's sort of become very popular in more recent times, which is NGS, which is your sort of needle in a haystack challenge, as we discussed earlier, because the metagenomics data that you can get is a lot, but a lot of it is quite irrelevant, especially when you're looking at pathogens. So a lot of it is human or host DNA or RNA, when you're really only looking for a tiny, that tiny needle in the haystack approach. And this is where ddPCR comes in. So it's really a powerful tool in rare event detection. So when we're talking rare event detection, there's two sort of splits when it comes to rare event detection. There's rare sequence detection, which is your analysis of a sequence in the high background of DNA. So you're only looking at that specific sequence. So maybe you're looking at a pathogen in human, so your host, which is like for environmental studies or microbial genomes or viruses in a host's presence, or you're looking at rare mutation detection. Now, in a rare mutation detection, something like a cancer mutation, you're looking at both the background and the sequence. You want to know both of those in quantity because you want to measure the wild type versus the mutant type. So those both can be utilized for ddPCR, and really helpful tool to monitor both. Now, moving on to some examples of pathogen sort of monitoring with ddPCR and where it's being used effectively. The first is monitoring of residual infection in HIV. Why is this being done? Well, it's critical for controlling the viral dynamics in a patient. And the problem is that the existing therapies result in suppression of the viremia particles below a level of detection that the current commercial assays can detect, and they have high measurement variability. So the solution is to use a published probe assay with the ddPCR format. Between 50 to 20 times improvement is what they detected in precision and accuracy when compared to the current commercial assays that they've been using, that they're currently using. And the limits of detection was down to 0.7 to 3 viral targets per million cells. Now, if we look at HIV infections, they continue to be obviously a very significant health risk. And these therapies that exist like ART, reduce the viral loads to levels that are quite difficult to measure with any of the current technologies. So it's important for us to be able to measure viral presence as low as possible and to monitor disease progression as your therapy goes on. So in any individual, the viral strains will vary and are affected by the course of the therapy. So having a ddPCR method that allows a much higher detection really helps in these sort of cases. So you can see here this in the slide, this proviral HIV DNA, so that POL gene was used to measure cellular viral reservoir size and long terminal repeats for viral dynamics. So both assays showed a significant increase in performance when used in a digital PCR format. And endpoint ddPCR was much more sensitive in detection of the mutants. And in this example, I think, yes, over 300 clinical samples were used in the study. So they used an existing TACMan probe assay without any modification. So they're an existing qPCR assay, and then they used the results to compare between qPCR and ddPCR. And you can see the results in digital showed a 5 to 20 times improvements in precision and accuracy. And the curve showed excellent linearity in concordance with measured and expected levels. And this was done on a QX200 system as well. Now, as we touched on in the last slide, that a major problem or barrier to conducting HIV cure research is the lack of accurate assays to quantify replication competent reservoirs across the subtypes across the world. So obviously in HIV, the reservoir contains latently infected cells that persist even during ART treatment, and these cells can be infected with HIV and carry this proviral DNA copy of the HIV genome, and it's stably integrated into the host cell chromosome. So the process of generating this proviral DNA through reverse transcription is error-prone, and most integrated proviruses contain mutations, insertions, or deletions, rendering them unable to produce new infectious virus. The remaining cells that contain the full-length intact proviral DNA and produce replication-competent virus are the source of viral rebound upon treatment's cessation and are the major barrier to cure. So basically, for many years, I think the gold standard for confirming that the reservoir is still intact was the quantitative viral outgrowth assay, and they rely on stimulation and culture of replication competent virus. However, they're quite labor intensive, they're low throughput, and they often underestimate the replication competent reservoir size of HIV. So here we're looking at an assay called the IPDA assay, so intact proviral DNA assay. And in this assay, they detect multiple targets across the HIV genome in a multiplex ddPCR reaction. And in this assay, they are much higher throughput than full-length sequencing with NGS, which can obviously be very labor-intensive and quite slow. And this assay was shown to work across subtypes A, B, C, D, and AE, and also able to detect down to a single copy of intact provirus. So really high levels of, really low levels of detection as well. And this is another one with HIV. They were looking, this case study was looking at very early in ART treatments, infants can alter the establishment and the long-term persistence of HIV infection. So they needed some sort of ultra-sensitive test to evaluate the persistence of HIV infection in a child through the age of 30 months during the therapy and after the therapy was discontinued. And for this, they turned to ddPCR because of the sensitivity of this sort of technology as well. So This is just showing you the comparison of the results, and the ultra-sensitive test showed very low but measurable HIV DNA at 24 and 26 months, and this was not detected by standard qPCR or real-time PCR. And there were 3 types of tests done at age 24, 6 months after cessation of the ART. In 1, the plasma viral load was detected to a single copy of RNA, and 2, a co-culture assay which could not find any virus competent to replicate itself. And in number 3, the 37 copies of HIV DNA per million was detected with ddPCR. So it really just shows you again the detecting level of ddPCR as well. And here, this is a totally different sort of application, but this paper was taken from ATCC. And I highlighted this specific paper because they demonstrated the lack of any viable diagnostic test that they have for babesiosis, specifically for distinguishing strains. So they wanted to see if they could use ddPCR and comparing it to qPCR and to see the difference. And this is what the outcome was. So they concluded our methods could be used as a sensitive approach to monitor the progression of parasitemia in rodent models of infection, as well as serve as suitable molecular tests and blood screening. Current methods of detection is blood smear microscopy. They could detect at day 3 post-infection in ddPCR. And if you compare that to qPCR, as you can see on the slide, that they still couldn't have any detection in real-time PCR at day 3. So just highlighting again that higher sensitivity of ddPCR when compared. This is from a article coming out of Cape Town. So in this study, they're looking at bioaerosols of a size of less than 0.1 micron can be generated by speech, sneezing, coughing, etc. And they looked at a quantitative measurement of the TB bacillary concentration in aerosol, and it may help to assess infectivity. They wanted to gain further insights into the airborne phase of TB and establish the bacillary concentration in exhaled bioaerosol. So here you can just see the different aerosol sizes and infectivity of those aerosol sizes with CFU units. Then when they placed the patients in a sealed room and they monitored the parameters like aerosol particle size, CO2 concentration, cough sound monitoring, and TB bacteria sampling devices. And then they showed that they had 35 patients that they had positive as TB positive, which they used the gene experts to detect this. And then they sampled the air for these. So they just looked at their various filters that they put in the room. They sampled at 28 liters per minute for 10 minutes. And then the mean was 2.5 CFU for each patient. So here you can see how much the sites that only the ddPCR was able to detect these aerosolized TB bacteria that was present in these patients from just sitting in the room. So quite an interesting study as well. Now, if we turn to quite a different sort of industry, we're looking at bacterial pathogens in poultry, in the poultry industry. So the traditional methods used in this industry is the culture method, and it's obviously quite time consuming. It's ineffective at selectively, it's a bit time consuming, obviously. And so they wanted to see if qPCR could compare. So they used it to compare to ddPCR as well as culturing methods. Obviously, keeping in mind that real-time PCR requires proper standards and was dependent on assay efficiency. For this, they used scalder tanks and chiller tanks for chickens, and they targeted Salmonella and Listeria and some other pathogens as well. And they wanted to compare at a low level and overcome inhibitors that are obviously present in these samples, and they wanted to have something that had higher throughput. So as you can see here in their results, ddPCR yielded a more precise and sensitive quantification of bacteria, ribosomal RNA in different types of samples. And you can see the SME, which was the start, the middle, and the end of the day was when they sampled these sort of samples as well. And you can see how in these results, the ddPCR showed a significant improvement for quantification for sensitivity. And then that same study looking at the different pathogens, they looked at practical use of ddPCR, qPCR, and culture, and you can see in often most of the cases, culturing was unable to detect, and real-time PCR was only able to detect in some cases, whereas ddPCR was able to detect in all cases. And in this example, we're looking at virus quantification and detection in terms of Zika and Ebola. So they basically just looked at these two infections. Obviously, these viral infections can ebb and flow during the seasons, and they can have a really intense and rapid outbreak, such as the case of Zika and Ebola. And during these outbreaks, it's obviously very important to manage the spread and the treatment of these viruses. So they need to check, they did a test to obviously see the assays that we run with ddPCR, and these are assays that we offer ourselves, and which you can see offered quite high levels of sensitivity. Even when they did a 10-fold dilution of synthetic Zika RNA, you can see how it detects at 5,580 copies per microliter, and in 10 it's 523, and 52.6, and 6.4, and 0.48. And you can see how linear the detection is in a 1 in 10 serial dilution over the sample. So obviously you're expecting 580, you get 520, so really accurate when compared. And then for the Ebola example, they were just showing you the effect of using a gradient PCR to show you the robustness of the assay, when just to show you that even on a not so well optimized assay, whereas obviously in qPCR, if you have a not well optimized assay, you can't rely on any of your results because your R squared and efficiency are wrong. Whereas in the case of with ddPCR, even though your assay isn't so well optimized, as long as your positives and negative samples can be separated, you will still get a fairly consistent copies per microliter read. Just because it needs to count positives and negatives, it makes no difference on the division between positives and negatives as well. So those were just some of the examples of pathogen monitoring and detection. So as you can see, in ddPCR, they really offer, with those benefits of the absolute quantification method, so not having to use any standard curves, not having to have standards included, which is obviously very expensive, very time consuming, and difficult to run in triplex. There's also the benefits of a much higher inhibitor tolerance and a much higher sensitivity. So in so doing, ddPCR really is a really good tool for outbreak surveillance, as we can see, being successfully used in wastewater-based epidemiology, but also in pathogen monitoring for hard to detect or limited quantities. Really a great tool for outbreak and pathogen monitoring as well. And just to review, the technology that we have, those are just some of the consumables that are part of the process. It's fairly standard. If you're looking at PCR, real-time PCR, you'll be familiar with almost all of these. It's basically plastics, assays, which is just primer rod probes, your oils, which is different because obviously you need something to partition with, and then your cartridges, which are the thing to partition on, and then you've just got your standard plates and tips, etc., which is standard for real-time PCR. So very familiar workflow as well. And then we have obviously a full portfolio with assays, reagents, and various sort of offerings. We also have design tools if you are currently looking to design your own assay. Maybe you're looking to do copy number determination or mutation detection, or genome edits, we have a tool that we can make use of alongside you guys, and we can design a full assay for you to run these sort of tests, very straightforward and quite easy to understand as well. And then just to highlight the support structure, obviously in the region, obviously we are distributed by Lasec, who's our partner, who Michelle is part of. They have a full team of application specialists, service engineers, sales representatives. And then obviously us at Bio-Rad, we have a full team of field application specialists, multiple throughout the region. We have regional specialists. We have a dedicated digital biology center that only focuses on digital biology, and obviously product managers and a dedicated technical support team. And all these are fully available to any of our customers. So any sort of support questions, we try to answer in the shortest time possible and always offer the best support that we can. I hope that gave you some more insight into ddPCR in terms of outbreak surveillance and how it can be utilized in terms of various applications, where we discussed wastewater monitoring, HIV, TB, some local, some pathogen monitoring, environmental samples. And I'm going to hand over to Michelle. And if you guys have any questions, please feel free to pop them on the chat and we can answer, and hopefully we can help you out with those questions. Thank you so much for your time.